Hello students! The topic for this week is about cellular respiration, or the process by which cells metabolically convert sugars into energy. To put in a bird's eye view, we will be discussing the pathways of the cellular respiration, the stages of cellular respiration, fermentation, anaerobic harvesting of energy, and the relationship among the different pathways. Before anything else, let us decipher between the process of respiration and fermentation. Initially, each process involves the usage of glucose as the raw material to commence the process. Moreover, the two process involves the process of glycolysis, producing byproducts like the nicotinamide adenine dinucleotide or NADH, a reducing agent, and the adenosine triphosphate ATP, producing another metabolic product known as the pyruvic acid. Now, when the pyruvic acid is successfully produced by the two processes, a different story revolves around its next fate, process dependent. In the respiration process, the pyruvic acid will proceed to the Krebs cycle until it reaches the electron transport chain. On the other hand, the fermentation process will involve the pyruvic acid, or its derivative, to form fermentation end products. The step-by-step process involved between the two will be thoroughly discussed as we move along to a deeper discussion. Cellular respiration, harvesting of energy. This slide connects the importance of photosynthesis and cellular respiration. That is, the photosynthetic reaction is the causative of the production of the raw material glucose to fuel the metabolic process, thereby creating energy in the long run. Notably, the end products like carbon dioxide, oxygen, and water are also recycled. Cellular respiration provide energy for life. The entirety of the process, though complex, is vital and crucial for an organism. The process of cellular respiration is a set of metabolic reactions to convert chemical energy from nutrients into ATP or adenosine triphosphate. The process occurs in the mitochondria of the cell. With this, you might already have an idea why the mitochondria is the powerhouse of the cell. As previously mentioned, the overall process of ATP production starts with the organic compound, glucose, C6H12O6. With the addition of oxygen molecules, the glucose will proceed to form carbon dioxide, water, and ATP. Cells tap energy from electrons falling from organic fuels to oxygen. The energy necessary for life is contained within the arrangement of electrons in chemical bonds within organic molecules. Now, an important question then is, how do cells extract this energy? The key to answering the question is understanding the redox reaction or the oxidation-reduction reaction. Note that in a redox reaction, once an electron is lost from a substance, an oxidation process occurs. While an electron is added to a substance, a reduction process occurs. Therefore, a molecule is oxidized when it loses one or more electrons, and it reduced when it gains one or more electrons. In this certain example of a redox reaction, in the cellular respiration process, the carbons in the glucose becomes oxidized while the oxygens become reduced. The stages of cellular respiration. The overall process of cellular respiration occurs in three major steps: glycolysis, pyruvate oxidation and citric acid cycle or the Krebs cycle, and the electron transport chain and chemiosmosis, respectively. This is the overview of the process. At this point, note the importance of the outer and inner mitochondrial membrane, the membrane infoldings, and which among the processes occur outside or inside the organelle. This slide is another depiction of the overview of the cell respiration process. Note that only pyruvate is allowed to enter the membrane of the mitochondria. Therefore, the initial process of breaking down glucose is essential to allow it penetrate the inside of the mitochondrion. We can say that glucose may be bulky to traverse the mitochondrion membrane, so it needs to be broken down to make it smaller for efficiency purposes. Now, how is the cell able to create ATP? ATPs are generated through 1. Substrate level phosphorylation, or 2. Oxidative level phosphorylation. The substrate level phosphorylation includes the direct transfer of a phosphate to an ADP or adenosine diphosphate from another molecule. Majority of the processes in the glycolysis and Krebs cycle involves substrate level phosphorylation. On the other hand, the oxidative level phosphorylation uses the ATP synthase and energy derived from proton H+ gradient to make ATP. This process is very much evident in the electron transport chain. Stage 1. Glycolysis. Note: glycolysis happens in the cytoplasm of the cell. It is in this process that the glucose is broken down into two pyruvate molecules. Stage 2. The citric acid or Krebs cycle. The Krebs cycle happens in the mitochondrial matrix. This process involves the oxidation of the pyruvate to a 2-carbon compound. And lastly, stage 3, electron transport chain. The electron transport chain involves the carriers NADH and flavine adenine dinucleotide or FAD. This protein-associated coenzymes shuttles the electrons to the ETC or the electron transport chain embedded in the inner mitochondrial membrane. It also involves chemiosmosis, and it generates ATP through oxidative level phosphorylation. In glycolysis, it harvests the chemical energy by oxidizing glucose to pyruvate. Moreover, in glycolysis, literally, the glucose molecule is cut into half, producing a 2,3-carbon molecule known as the pyruvate. Moreover, 2 NAD+ molecules are reduced to 2 molecules of NADH. A net of 2 ATP molecule is likewise generated. But later, you will see that the ATP will just be invested in order for the process to proceed. Note in this slide the addition of 2 phosphate molecule to 2 ADP, producing 2 ATP molecules, while 2 NAD+ will gain electrons, thereby reduced, forming 2 NADH molecules. ATP is formed in glycolysis by substrate-level phosphorylation, during which an enzyme transfers a phosphate group from a substrate molecule to ADP. The steps of glycolysis can be grouped into two main phases. In steps 1 to 4, or the energy investment phase, energy is consumed as 2 ATP molecules are used to energize a glucose molecule, which is then split into two small sugars that are now primed to release energy. We can see in this process that it invested the ATP in order to fuel the succeeding process. Therefore, no net ATP is gained in this process as it will be reduced. However, in steps 5 to 9, the energy pays off when two NADH molecules are produced for each initial glucose molecule and ATP molecules are generated. This is a slide showing the first step or the energy investment phase. Steps 1 to 3, a fuel molecule is energized using ATP, and that is glucose. So if we can see it step by step, from the initial glucose molecule, an ATP is invested in order for the glucose to be converted to a glucose 6 phosphate. Note the insertion of the phosphate molecule at the 6th carbon molecule of the glucose compound. 2. Glucose 6 phosphate will be converted into fructose 6 phosphate. That will just involve some modification or rearrangement in the carbon molecule. By step 3, another ATP is invested in order for the fructose 6 phosphate to be converted to fructose 1,6-bisphosphate. Note the addition of another phosphate molecule in the first carbon of the fructose 1,6-bisphosphate. From this fructose 1,6-bisphosphate, it will be primed or cut into 2-glyceraldehyde 3-phosphate or G3P. For the remaining of the steps, the energy pays off. At step 5, a redox reaction generates NADH. That is, from the glyceraldehyde 3-phosphate, another phosphate molecule is added that will now harvest 1,3-bisphosphoglycerate. From the remaining steps, 6 to 9, ATP and pyruvate are produced. From the initial 1,3-bisphosphoglycerate, ATP is yielded back in order to create 3-phosphoglycerate. From the 3-phosphoglycerate, there will be another round of rearrangement of the molecules for it to become 2-phosphoglycerate. That is, the phosphate compound or the phosphate molecule will attach to the second carbon of the 3-phosphoglycerate. Following the next step, from 2-phosphoglycerate, it will be converted into phosphoenol pyruvate, PEP. And then lastly, from the PEP, ATP will again be regenerated and it will now yield the desired end product for this process, which is the pyruvate. Glycolysis harvests chemical energy by oxidizing glucose to pyruvate. This is the general equation of the overall glycolysis process. For glycolysis to continue, NADH must be recycled to NAD+ by either 1. Aerobic respiration occurs when oxygen is available as the final electron acceptor, and 2. Fermentation occurs when oxygen is not available. An organic molecule is the final electron acceptor. The fate of the pyruvate depends on oxygen availability. When an oxygen is present, the pyruvate is oxidized to acetyl coenzyme A, which then enters the Krebs cycle. But without oxygen, the pyruvate is reduced in order to oxidize NADH back to NAD+. We can see here the interplay of the aerobic and the anaerobic process. So as mentioned a while back, when there is oxygen, the pyruvate will be or will proceed to the Krebs cycle. While if there is no oxygen, it will proceed into two different steps. First, NADH will be converted to NAD+ to form lactate, or the pyruvate will be initially converted to acetaldehyde with the release of carbon dioxide in order to yield ethanol. The transition reaction oxidation of pyruvic acid will now be discussed by Sir Noel Galido. Thank you.